So today we are going to start the microbial growth. Am I audible? Hello?
Yes, ma'am, you're audible.
Yes, ma'am, you are audible.
Okay. So today we are going to learn about the microbial growth. So I think many of you are already accustomed with microbiology, but still I am going to just give a brief description that who all belong to this group, like members of the microbial world. So we have here the bacteria, archaea, and the eukarya. And when we study these microorganisms, we call this domain as microbiology. So today we'll mainly study the microbial growth. So firstly, I would say that what does growth means to us? It's simply that increase in cellular constituents, like in rise in number, or it will be change in cell shape and size, etc. So for microbes, like most of the prokaryotes, we know that they reproduce by binary fission. So what is this binary fission? That is, this is the mode of division they follow. Like I can say here, this is the parent cell. Okay. So this parent cell prepares for division. Okay. So they enlarge, the DNA gets duplicates and the membrane, etc. Then the cell begins to divide. That is, a septum is created here, and then they are divided into two daughter cells. Okay. So this is the main mechanism of binary fission. Now, as we are going to study the microbial growth, so I would like to say that how this growth is studied in lab. So in lab, what we do is that we have some microbes, that is the inoculum. Now we inoculate this in a fresh growth medium, and then we incubate this at 37 degrees Celsius for 24 hours. And then we measure the OD values, that is the optical density values, at respective time intervals. And now what we do is this, after taking the OD values respective time, then we are going to plot this. Okay, plot this in a graph. That is, the growth of microbes can be plotted as a logarithmic of the number of viable cells versus the incubation time. And the curve that we'll get, it is called a growth curve. Now the curve looks like typically like this. Okay. The curve looks typically like this, with the y-axis having the number of viable cells in logarithmic form, and the x-axis have the time. Now here we have is that several phases. Like this is the lag phase, then we have the log phase, then stationary phase, and then this is the death phase or the decline phase. So what is the lag phase? Now for any cell, like microbial cell, when you are inoculating a previously cultured cell to a fresh medium, they need to adjust themselves to the new environment. So that is the period called the lag phase. Okay, the microorganisms, they prepare themselves for the division. Then there can be necessary for some essential cofactors, or their ATP must be maybe depleted, and then synthesizing a few essential things. Okay, so they would need some time. So that time is called the lag phase. Now this lag phase can vary according to microorganisms. Okay, like maybe they can have a very short lag phase, like if it is a very young culture, and if it is a very old culture, now this culture you need to actually culture next in the fresh medium, you will need a longer time, so the lag phase may be a longer. So this, after stabling in lag phase, the microbes move on to their exponential phase or the log phase. Now this is the phase when they are at maximal growth rate. Okay, they divide at constant intervals. Now they also produce the enzymes and proteins, etc., which are needed. And now this phase is also called a trophophase or an exponential phase. The primary metabolites that we get are produced here only, and we can study the mechanisms of bacteria about any applications from this log phase only. So as we are doing a lab culture. So the growth is studied in a batch culture, like we are providing all the nutrients at a time and then we are starting it. Okay, it is in a closed system. So after a certain time, the what to say that the nutrients will actually get depleted, and so the microbes will enter a stationary phase. Okay, that is the total number of viable microorganisms will now remain constant. Okay, so there will be a balance between cell division and cell death. So why does this happen? This is the question, that why does this happen? There are two reasons mainly. That is, when an essential nutrient is used up, that is, it is in limited amount. So when it is used up, there will be a less growth, or the growth will cease, and accumulation of waste, which turns very toxic to the microorganisms, and maybe they are still living but they won't divide. Okay, and that's the reason that the cell division and the cell death becomes in a balanced nature. Okay, and the viability remains constant. And after a certain period of time, this phase will also turn into the death phase. Like, even after you shift or transfer the inoculum from there to any fresh medium, maybe they won't divide anymore. Okay, so this is the death phase. So this is a typical growth curve of bacteria, and I'm just giving a very brief description about it. I'm not going into a very depth of it, as this is the first class. So now we come for some of the equations. Like, as we have studied the graph, now we need to know that how much time a bacteria needs to grow, or how many generation does it needs. Okay, so all these things are actually studied. All these things are studied in the log phase. Okay, so in exponential phase, the microbes divides at constant intervals. And here is the thing we actually go for all the studying things. Okay, so the population will divide in a number during a specific length of time. And this time is actually called a specific length of time. And this time is called generation time, or the doubling time. Okay, so as the population is doubling in every generation, so there will be an increase in population, which is denoted as like 2 to the power n. And now we have to go for the equations, like the state that we have some initial population, which is NZ, denoted as N0, final population denoted as NT, and number of generations we have N. Now the equation goes like NT is equals to N0 into 2N. This is the final population, which is equals to initial population into 2 to the power n. Okay. Now for solving this, we take log on both the sides, and we get an equation like log nt minus log n naught divided by 0.301. This is equation 1, which we can use for solving the problems. Now we have the second equations, like mean growth rate constants. Now what is this? The rate of growth during the exponential phase in a batch culture can be expressed in the mean growth rate constant. Okay, so what is this? Like, number of generations per unit time. Okay, in each time interval, how many generations can be expressed? Okay, so this is k is equals to n by t. N is the number of generations, and this is per unit time. Now from the previous equation, we already know that what is the value of n. Here we have solved with taking log from both the sides. So we have got one equation here. Now what we are going to do is like put that equation, or substitute that equation in place of n, and we get that log nt minus log n naught divided by 0.301 into t. This is for equation 2. And next we come for the mean generation time. Here we get that the time taken for the population to double in size. Okay, mainly we go for the time here. So this is g is equals to 1 by k. So here also we have the k value here from the previous equations. Now we are just going to substitute that value here, and we'll get t by n. This is the equation 3. So mainly these 3 equations are needed to solve the preliminary questions of growth. And I think GATE gives this type of questions only. Okay, from the previous papers that I've seen, or I've attempted the GATE, I've seen these questions are very preliminary questions from growth. Okay, so these are the equations you will need to solve it. So I would go for one numerical here. I think so this was in GATE 2010 or 8, like that. So this is a very preliminary question. Like you can also try, all the participants, just you can write it down. A bacterial culture had an initial cell density of 10 to the power 3 cells. Okay, 10 to the power 3 cells per ml. In 6 hours, the cell density reached 10 to the power 6 cells. Now the formula is also given. They are providing the formula also. So we have to find here what is that, the number of generations. Okay, so these are the options given here. So we have the initial cell number as this, final cell number as 10 to the power 6. The time is given that 6 hours. Now as per the formula, we know that n is equals to log nt minus log n naught divided by 0.301. We are just going to put these values, solve the equation, and we will get this answer. Now the answer we have got may not exactly match with the options that are given here. So we have to find the best suitable one from here, which is the nearer one. So here 9.9, that's equals to 10. So we are going to opt for this option. Okay. We come for the second numerical. That is, the generation time for E. coli is 30 minutes. For an exponentially growing culture, the initial number of bacteria required to reach a number of 10 to the power 9 in 24 hours. So what is the question asked here? Is like generation time is given 30 minutes. Now in an exponentially growing culture, they have asked the initial number of bacteria that will be required to reach the final cell number of 10 to the power 9 in 24 hours. So basically everything is given here. The generation time is given, the final cell number is given, and also the time is given. So generation time is 30 minutes, final cell number is 10 to the power 9, and the time given to reach is 2 hours. So here we are going to convert the hours into minutes here, that is 120 minutes. And now we are going to calculate the number of generations. So number of generations we can calculate by T by G, that is 120 divided by 30. That this equation we already know that G is equals to T by N. From here we are going to substitute and we're going to get the number of generations here. And now we are going to calculate for the NT, that is the final cells, final population. So we'll get 6.25 into 10 to the power 7. Okay, this 10 to the power 7 is already given, and you have to solve it and bring the value in this 10 to the power 7 value only. Okay. Yes.
मैम प्रीवियस न्यूमेरिकल में डाउट है मैम, वो टाइम 120 मिनट्स कैसे हुआ?
ये 2 आवर्स है, तो हमें अगर इसे मिनट में कन्वर्ट करना है, क्योंकि ये जनरेशन टाइम मिनट्स में है ना. ओके
मैम, क्वेश्चन में 24 आवर्स पूछा है ना?
Oh, sorry, it was 2 hours. Sorry. This was 2 hours only. My mistake. Okay.
Okay.
Hello? Yes, the question was 2 hours only. I'm sorry. Okay.
Okay, ma'am.
Okay, even if it is 24 hours, then it will be 24 into 60 minutes. Okay.
Yes.
I'm sorry. So next, the third one. So here I have this a growing culture of bacteria with a doubling time of 20 minutes, and it reaches the cell density of 2 into 10 to the power 8 in 3 hours. Now the question is asked that how much time it would take to reach the cell density of 1 into 10 to the power 8 cells per ml. Okay, so doubling time is given, that is 20 minutes. Final cell bacterial load is also given, that is 2 into 10 to the power 8 cells. Now time is also given, that is 3 hours. Now we are changing this time into minutes. 3 into 60, it's 180 minutes. Now if 2 into 8, 10 to the power 8 cells in the cell that takes 3 hours, then how much time will 1 into 10 to the power 8 cells need to reach this volume? So it will be just 3 by 2, which is equals to 1 hour 30 minutes or 90 minutes. And here goes the answer, 90 minutes. So till now we are reading about studying about the batch culture. So actually I didn't go to the very depth. I was just giving you a brief description of it. So in lab, lab scale we can study for the batch culture, like all the medium we are putting together, and then we are studying the growth pattern or other things. But in industries or large scale, we need to go to maintain that exponential phase. In batch culture, what happens after a certain time, the microorganism shifts to a stationary phase. But if we have to culture it and apply the microorganisms in our daily lives, like all other things in industries, factories, food factories, etc., we need to maintain them in the exponential phase. Now how do we do this? So here comes the concept of continuous culture of microorganisms. Now what is this continuous culture? So this continuous culture is that continuously we are maintaining the environmental conditions. We grow the microbes with the constant environmental conditions, like supply of the nutrients, removal of the waste, etc. Okay. So there are two major types of culture systems, which is we know that chemostat, and second one is the turbidostat. So this is the typical structure of a chemostat. Now here we have the media reservoir, okay, where the fresh media, sterilized media is there. And this is the chemostat vessel or the culture vessel. Now this media is continuously being supplied here, also with the oxygen, and continuously there the effluents, the eluents are actually collected. Okay, so waste and also the whatever we need, the microorganisms with the media. Okay, so in chemostat, fresh media is fed at the same rate as the media containing the microorganisms are removed. The culture media contains a limiting nutrient. So this is the main thing in a continuous culture. In batch culture, there is no limiting nutrient. The nutrients are already given. Now they will deplete. But here we have a limiting nutrient, which is called an essential nutrient. This is needed in the continuous culture to actually keep them in the exponential phase. Okay, because of this, the growth rate is determined by the rate of which the new media is fed. And the final density of the cell depends upon the concentration of the limiting nutrient. The rate of nutrient exchange is expressed as the dilution rate, and we actually calculate it as the flow rate divided by the culture volume. Now, flow rate is how much media is flowing from this reservoir to the culture vessel, and the volume of the vessel. Okay, so we have a numerical here for the continuous culture. A continuous bacterial culture carried out in a chemostat is set to a flow rate of 40 ml per hour. Okay, it's 40 ml per hour. The culture volume is equivalent to that of a cubical container having 10 centimeter size. Now they have asked for the dilution rate per hour of this system. So flow rate is given 40 ml per hour. Volume is also given, but in a bit different way. That is, it has said that the volume is equivalent to that of a cubical container which is having 10 centimeter. So from here we have to actually find the volume, that is 10 cube, we'll get 1000. And now the dilution rate with the equation flow rate divided by the volume, that is 40 divided by 1000, which is 0.04 per hour. Okay, from here we actually come for the monokinetics, that is for the chemostat mechanism. Now at very low dilution rate, as I have already spoken about the dilution rate, we come to know that at very low dilution rate, and increase in D causes a rise in both cell density and also the growth rate. So here we come for the monokinetics, and it is same as the enzyme kinetics we have, describes the effects of a substrate concentration, that is the limiting substrate. And the equation goes here for like mu is equals to mu max into S divided by Ks plus S, where we have the mu max equals to maximum specific growth rate, Ks equals to substrate specific constant, S is equals to substrate. Okay, taking the reciprocal of this equation, like the LB equations, whatever we do in the enzyme, that is 1 by mu is equals to Ks plus S divided by mu max into S, and we get the equation as Ks divided by mu max into 1 by S plus 1 by mu max. Here this is the slope, This is straight line graph equation. This is the slope. This is the x-axis, and this is the y-intercept. We come here for the graphical representation. This is the same graphical representation as the Michaelis-Menten representation. Yes, the same one. Only we are going to use this for bacterial growth. That is, here we have the half of mu max, which is Ks, and this is called the Monod constant. This is the graphical representation of Monod's equation, and here also the reciprocal of it, I mean the straight line equation is the hyperbolic equation, and this is the straight line equation we have, that is 1 by mu, and x-axis is 1 by s. Okay, these are the graphical representations. Though I don't think so questions come in gate from Monod's equations in microbiology. I haven't seen much questions comes, maybe theoretical, but never the numericals. Okay. So next we have the turbidostat. This is a typical structure of the turbidostat. This is the sterile reservoir of the media, and here is the culture vessel. This is the culture vessel, and this media flows here into the culture vessel, and we have a photocell which actually detects the turbidity here from the light source. Okay, so the difference between the chemostat and the turbidostat is that in chemostat actually we determine the growth rate by the limiting nutrient, and in turbidostat we go for measuring the turbidity. Okay, so it has a photocell that measures the absorbance. Yes, has a photocell that measures the absorbance or turbidity of the culture, and the dilution rate in turbidostat varies. Culture medium contains all the nutrients in excess, and this works at a high dilution rate. So, okay, bye, good night.
Thank you.
Yes.
Bye, good night.
Yes.